welcome everybody to the final Idaho Soil Health 5 for 5 of this winter season. We are so grateful to our speakers for joining us today and for the audience here to listen. My name is Lucille Rollins, and I'm a soil health specialist with the NRCS based here in Boise, and am joined today by my co-facilitator, Courtney Costin, who is the University of Idaho Extension soil health educator. So we have four really knowledgeable speakers today delivering 5 slides in about 5 minutes on the topic of the connection between soil and human health. And those speakers are Dr. Stephan Van Vliet from Utah State University, Laura Moore from Colorado State University, Jade Koyle from Ancient Grains, and Rem and Karman Fox from Mad Fox Farms. So as we traditionally do, we'll have a short Q&A segment after each speaker. So please feel free to drop your questions in the chat or come off mute during the Q&A segment to ask them directly. We would love that. And a reminder that today's session is recorded and it will be available later on at the University of Idaho College of Agricultural Sciences YouTube page. So with that, Dr. Van Vliet, I'll let you take it away and get us started.
All right. Thank you so much. Could you give me a thumbs up if you can see this? All right. Perfect. Then let me get started. So it was hard to condense everything into 5 slides, so I did my best. I'll give a little snippet about the work that we do. So I'm the director of the Center for Human Nutrition Studies at Utah State University, and we have a strong interest in linking soil, plant, and animal health to nutrient density, particularly looking at linkages across that. Perhaps empirically, a lot of producers may experience this when soil health and the health of then this case their forages or their human edible crops would improve, that they see an improvement in animal health. And over time, I've come to the belief, or at least supported by our data, that plant health and nutrient density are probably interchangeable, and that animal health and nutrient density are probably interchangeable as well. That if you have a healthier animal, it's a more nutrient-dense animal, and vice versa. So within our lab, we have two major interests. We have a strong interest in linking soil, plant, animal health to food nutrient density. We use a lot of what is called metabolomics. These are advanced mass spec-based techniques where we profile samples really beyond just the surface level and study a lot of different compounds in there. And then we also have a clinical facility where we feed people foods in randomized controlled trials to determine potential health effects. Over the last few years, we've conducted a series of studies. Most recently, we finished a study where we compared pasture-finished animal-sourced food consumption in a 16-week trial in 80 people versus more conventional feedlot-finished and CAFO raised animal-sourced foods, and actually found that we can meaningfully raise the omega-3s in the blood of people by consuming land animal-sourced foods as well. So in the first part of our studies, or at least the metabolomics profiling, we believe there's especially in the current times, there's a need to profile beyond protein and fat and just consider those surface level foods, right? Because we have unfortunately dumbed down foods to protein and fat, and call soybean protein and meat protein, and call avocado fat and an almond fat. But these food sources contain thousands of nutrients or thousands of metabolites that are capable of impacting our metabolism. And it's also those particular metabolites that are often impacted by the way that we grow. Earlier on, we were talking about how healthy plants can ward themselves off from insecticides. It's just because of production of plant secondary metabolites that are things like polyphenols and flavonoids that also give flavor and health to our the crops that we eat, and also improve the health of our animals. So the USDA Food Composition Database tracks about 150 components, but food sources contain an estimated 30,000 compounds capable of impacting human metabolism. And as you can see here from the Nutrition Facts panel, we cannot determine that. And it's quite hard to determine differences in nutrient density amongst food from the nutrition facts panel alone. So what we do oftentimes is we profile the food metabolome, look at hundreds or close to 1,000 nutrients. We then feed those to people. And we study the human metabolome, so the transfer of nutrients from the food to the human. And then also its interaction with the nutrigenome, which is a fancy way of saying how do these nutrients interact with our genes and our metabolic disease risk. So in a series of profiling studies that we did, we found that grass fat is in grain fat and grain fat is in grass fat. But what is particularly novel about this work is that we studied 800 metabolites, so much beyond just omega-3 fatty acids and protein and vitamins, and we found actually that there's a 56% difference in beef and a 52% difference in bison, so about 50% or so. Some metabolites are upregulated in grass finishing, some in grain. This does not per se mean that higher in here is good or bad for that matter. I mean, it depends. We look at a variety of metabolites in here, as well as oxidative stress markers on the animal. But there is a large difference that is probably larger than previously appreciated, and it's really interesting because these data, 50%, there was a really nice study a few years ago that found that about 50% of what circulates in our blood is directly related to the foods that we eat. So sort of a half-truth to the saying, you are what you eat. And it seems that if you finish an animal on two wildly varied diets for 120 days, you also get sort of this 50% difference. In our group, we've had a particular interest in what is called phytonutrients or plant phenolic compounds, because when you compare total mixed rations to more forest-based diets, oftentimes the difference in grass-fed and grain-fed beef is very similar. Maybe the grass-fed beef is a little bit leaner, but the protein content is fairly similar. The vitamin and mineral content is fairly similar. Maybe some minerals might be a little bit higher in grass-fed beef. Some B vitamins like B1 and B5 that are rich in grains are a little higher in grain-fed beef. But we're particularly interested in where these diets vary very much is that a total mixed ration provides energy, protein, vitamins and minerals, but it does not really provide as much depth in these phytonutrients, these 30,000 metabolites that are found within plants that are being grazed, and especially with diverse number of plants. So in here, in a little snippet is that we looked at a major phenolic compound, benzoate, and this gets transformed in the animal into hyporate, which is an antioxidant associated with decreased or improved metabolic health in mammals. It also increases in us. So we found that higher levels in forages, higher levels in grass fats. And this is my final slide. So within that work, we've been really interested in looking at understanding how these various herbs and shrubs and grasses and legumes and other potentially medicinal plants, how those phytonutrients within those plants get transformed within the animal, gets transformed within the ruminant, how they impact their health, because a lot of these have antioxidant effects, antimicrobial effects, they improve mitochondrial health, and then the transfer of those antioxidants into the meat and the milk, where they provide two major things. They provide flavor, because things like, I think a lot of farmers might be aware if animals consume certain plants that flavor might translate. If they eat plants rich in rosmarinic acid, you might have some rosmarinic acid in the meat, provides flavor. And then study their considerable health benefits to themselves and potential health benefits to humans. And those are my slides. And we do work a lot with farmers in this space as well. So I'm grateful for that collaboration, and thank you for attending.
Thank you so much, Dr. Van Vliet. There was a lot of really great stuff in your slide deck today. I'm just checking the chat to see if we have had any questions. So far, I don't see any. So I guess I'll ask the first question, which is, you said that you work with farmers. So you have a lab that farmers can send samples to. Can you talk about, I guess, what that that process looks like and what individuals can expect to get back as far as results and interpretations?
Oh yeah. So it actually started out of the research because we were publishing these findings, and farmers would contact us and say, "Hey, so this presentation, and can we submit those samples?" So I was like, "Sure, I think so. Why not?" And then we started to work on that. We did a large project together with the Bionutrient Institute called the Beef Nutrient Density Project as well. And but yeah, it's fairly simple. I mean. If you reach out to me, we have a variety of tests that we can do, and they get a nutrient density analysis. So it'll be fatty acids, minerals, protein, phytonutrients, fat soluble vitamins, basically all macronutrients and micronutrients and phytonutrients. We provide farmers with project reports back and their results in a spreadsheet. We're also working on a data platform online. And we benchmark that to other grass-fed producers, obviously anonymized, and grain-fed samples as well. And we also work on crops. In the interest of time, I just presented on beef. But yeah, I always jokingly say, as long as people don't send us rocks, I'm pretty sure we can extract it. And we provide them with the data back and interpretation as well. So that has grown a lot over the years due to just the natural interest. So yeah, if farmers are interested, can reach out to our group, and we'd be happy to profile it.
Okay, neat. Do you have a sense of how folks are using the data and the interpretations you provide them?
Yeah, at the moment, a lot of farmers are using it for consumer education. So they might have a like blog post or use it on social media. And I think a lot of producers are also uh, providing it or using it sort of for to confirm, right? A sort of a hunch that they have, and especially when we work with farmers or when I go on field sites, right? And you can see, okay, my crops I think are more productive, or I have, a longer, have a lot of soil cover, and I think, my carrots have a much better flavor than maybe what I buy at a grocery store, right? So farmers probably interpret that. Same with grazing animals. I have a feeling that they're producing healthier animals. So when we provide that data, it oftentimes also provides a confirmation for the farmer that they're on the right track. And I've had several farmers that submit over time as they implement and are longer in through their journey on implementing regenerative or agroecological practices to see if there's this consistent improvement. So,
Thank you. And we have had some questions come through the chat. So Shiloh asked, oh, uh, chat moved a little bit. Okay, so Shiloh asked, have you monitored the level of proteolytic bacteria in relation to animal proteins? And have you seen a reduction in the putrefying toxins produced by that process?
we have not per se looked at that aspect of things in terms of proteolytic bacteria in relation to animal proteins. What we've looked at within the animal protein or the meat in general is we looked at a series of oxidative stress markers. So these are things like 4-HNE and other compounds that, at least according to the World Health Organization, are maybe potential reasons why meat is a potential carcinogenic, but we actually found that those are strongly related to animal health as well. So by the way that you raise the animal, you can lower some of those levels of oxidative stress or some of the maybe things that may be considered detrimental. It's a very long discussion on the because that you can see that the unprocessed meat is not really associated with cancer, and so there's a little bit of discussion on whether we get high amounts of these carcinogens in. But you can see that those are strongly related to animal health. That we know, and you can lower those by producing a healthier animal.
Okay, thank you. And then for the good of the group, it looks like Brad had provided an answer, but for the good of the group, I'll share this question from Jade. Do you profile foods other than meat?
Yes, we profile pretty much any food. Yeah.
And Taryn asked, have you looked at other ruminants, for example, sheep?
Yeah, we have. We have looked at sheep, at goats. We've also just did some analysis. It wasn't funded, so we had to do maybe a more limited scope We looked at a lot of wild animals as well, deer, elk, caribou, nilgai antelope, grouse, etc. So yes, and typically what we see is that maybe sheep and goats that browse more shrubs and forbs, they actually get exposed to higher levels of phytonutrients. So their phytonutrient levels rise more than maybe in terms of grazers with cattle or bison. Although there's exceptions for sure, especially here out west, when some cattle might go for the sagebrush and gets exposed to high levels of phytonutrients as well. But in general, we found that the phytonutrient levels in sheep and goats, because of the way that they browse, as well as some wild ruminants, get further elevated compared to um, cows.
I'm curious, how far is that difference between, say, ruminants and cows when you look at cows, when you compare cows that are grain fed versus grass fed? Like how
So
big?
in grass fed, we see about 2 to 3 fold higher levels of phytonutrients, and then the on average, at least, the goat and sheep are about twice as high than cattle. And then when we profiled wild ruminants, they're further elevated. But when we look at individual data points, some grass-fed beef producers, so this is running in broad strokes, some individual grass-fed beef producers, especially ones that are grazing very biodiverse pastures, they may reach sheep and goat levels or more wild ruminant meat levels as well. So what we found is that typically that biodiversity, so the ability of the animal to select from a variety of different plants, typically has a benefit is associated with improvements in their health as well as the nutrient density of their meat. It's like one example, for instance, an analogy would be if we go into a buffet with 50 different foods, we're much better able to select or better able to nourish ourselves than maybe we go to a cafeteria and we get a plate with maybe the same 3, 4 staple foods.
Got it. Thank you. And we've had some more questions come through the chat. So Barry asked, how successful have your cooperators been able to monetize these results in their products?
I only from feedback from farmers, I can speak to that, is that they typically have leveraged the data to educate their consumers and to have tangible data that in many cases would indicate that their nutrient density, that show that their nutrient density is improved compared to a commodity product. So that's how they've been able to monetize it. I don't have exact numbers, but given the demand and also the fact that a lot of producers test yearly, I assume it's the cost is worth it.
All right, great. And then Annalyn shared, what are your goals this year as a lab and individually?
Well, great to see you again, Helen. We've talked and met a few times in the past as well on various conferences. So what we are launching in our lab, since we have a lot of producer submissions over the last year and this year as well, we're launching an interactive data platform where producers can log in and get their data sent to them through an interactive platform. They can click through, benchmark their samples against a variety of other foods even. Right now we're sending producers more of a PDF document with a report, but we're moving that over to an interactive data platform where it's kind of like if you get your blood work done at the doctor, you get your results, but in our case, you can also compare to other results. So that's been very exciting. We got a big Bison project coming up, and well, individually, I would say we have a newborn. So individually would be being the yeah, taking care of our newborn, which has been very, very enjoyable.
Wonderful. And then last question from Shiloh, is most of this data published and how would someone access the results of your studies?
Most of the data has been published. We always make it a priority to publish everything open access. Um, so yes, a lot of this data has been published. There's usually about a 1 to 2 year lag when we have the data versus when it gets published into peer review. I'm sure if there's other academics, they can appreciate that it takes a little bit of time. But I'll put my Google Scholar profile in the chat, and then I've also given plenty of webinars and recorded YouTube or YouTube recordings where we go deeper and present uh, to folks for an hour or so. So that might also be a good thing to look at. If you type in my name on Google or YouTube, there'll be various presentations on this data more in depth. People are interested.
People seem very interested in your work. You've got a lot, you've got a lot of content out there, which is awesome. So, okay, well, thank you so much again, Dr. Van Vliet. That was great. Learned a lot. And up next, we have Laura Moore from Colorado State. So, Laura, I'll let you take it away.
Hi, give me just 1 second to try to share this. Okay, is that okay?
Yep, it looks good.
Perfect.
Thanks.
Okay. Hello, my name is Laura Moore. I'm a PhD candidate at Colorado State University in the Soil and Crop Sciences program, working with Dr. Kelly Wright. And my work is all about using the soil microbiome to identify indicators of soil health and carbon processes. And this talk is going to be about translating those microbial functions into actionable insights for sustainable food systems and human health. So we've known for a really long time that microbes are playing these really important processes in the soil. They're doing everything from decomposing organic inputs that are contributing to climate regulation and greenhouse gas emissions. They're converting nutrients into plant available forms, promoting nutrient cycling, and that nutrient cycling is helping with promoting plant growth and stress tolerance. They're also capable of controlling pests and diseases and degrading pollutants in the soil. But it's only really been in the last decade or so that we've had the technology to really study the microbiome in a meaningful way. And so as we continue to navigate how to improve agricultural management practices to increase overall soil health, we're just realizing how powerful the microbiome is in terms of what it can tell us. And so the field of soil science has been utilizing 16S and ITS for some time now, which can give us information about which organisms are there and what abundance and how diverse they are. But these data can't really give us anything functional about as to what microbes are actually doing in the soil. And so now, thanks to advances in sequencing technologies, we have multi-omics, such as metagenomics and metatranscriptomics. And so we can now see exactly what genes are present and what organism has, what's actually being used, and what ecological functions those genes or metabolisms actually are encoding. So this means we can now see if an organism is able to do something like fix nitrogen or degrade cellulose, or if it can break down those heavy metals or pesticides in the soil. So this is really exciting because in human systems, these kinds of technologies are being used primarily in the gut microbiome to be able to predict or diagnose health concerns for people. And so we think that the soil microbiome could do something similar for agricultural systems. And so this opens up an entirely new world of understanding for us. Our lab is developing a national agricultural soil microbiome database that will compile microbial data from places like the Joint Genome Institute and CBI. We have a lot of collaborators and a lot of data from our own lab. And this will all get paired with management, climate, and soil physicochemical data into one collated resource. And then we have created on the far right, you'll see this annotation tool that is called DRAM-AG that can translate that information into something that can become meaningful. So we can start to see what kind of soil processes a soil is capable of forming. Like you can see that your soil sample is capable of actually doing nitrogen fixation. And so we've now compiled over 3,000 samples across the US, and we're going to start using machine learning to identify what features of the microbiome are most important for soil health metrics, and then begin to identify patterns of how specific functions change under varying climate and management factors. So our vision is that these kind of data can improve soil health tests and even begin to provide predictive information as to how likely management practices may work under specific conditions. And so I think it's probably helpful to walk through a example of what this can look like in practice. So we can create these biologically informed soil health tests using collected genes in a sample to tell you what your sample is capable of doing. And so if your sample has, let's say, an abundance of ammonium monooxygenase genes, which we know are the genes that are involved in nitrification, this could indicate that your soil is functionally primed to convert ammonium to nitrate. So in coarse textured soils where there might be irrigation or heavy rainfall, this could indicate a high risk of nitrate leaching. And so while a traditional soil health test can give you a snapshot in time of your ammonium or nitrate levels, the microbiome can begin to show you what your soil is capable of doing, even if it hasn't done it yet. So this kind of information can help inform management practices where maybe you want to instead use a split nitrogen application or consider using nitrogen inhibitors. So overall, as we begin to understand what the microbiome has to show us and figure out how to apply that information into something that producers and land managers can actually use, we hope that these data can empower our understanding of how to improve soil health and make more informed decisions on management practices to increase overall productivity and nutrient density of crops so we can continue to support human health and our ever-growing population. Yeah, thank you for your time, and please feel free to contact me. This is a QR code for my LinkedIn, and I also have a website and university email. Of course, there's a lot of people involved in this project, and we're really grateful to our funders. Thank you.
Awesome. Thank you, Laura, for that. Again, some great information. And I guess I'll start us off with a question. For context, the USDA launched this new program in December called the Regen Pilot Program. Soil health testing is a required component of that. In your slides, you're talking about the potential for, I guess it sounds like a new suite of soil health tests. How far off do you think we are from having these tests that you're talking about? And yeah, I'll start there.
Sure. I think we're going to be able to have something tangible in this next year. So our plan is to have the database and our annotation tool fully launched by this summer. And so at that point, we'll be doing a lot of testing as to seeing which features are really important. Because I think, in some soil health tests, you see like proxies of microbial indicators, things like beta-glucosidase is a really popular one. And that's just an example of like one enzyme that is doing this function, but there are hundreds, if not thousands, of different enzymes that could be doing similar functions. And we've kind of used beta-glucosidase because it's easier to measure, but we don't know if it's actually fully representative of the functions we are thinking it's performing. So I think there's going to be a lot of testing there to figure out what's actually relevant, and then figuring out how to apply those things to soil health tests or soil health factors that feel actually meaningful. So I think by the end of the year, we'll have a clear picture of what that looks like and how to apply it. But yeah, I think we're probably pretty close.
Okay, and so you're saying this summer, so folks could potentially send samples to you for analysis, or you're working with that set of 3,000 samples.
Sure.
And
So both. Our lab is setting up we are working on trying to get a program started where we can offer that as a service for people. So we're hoping to be able to launch that this summer. So TBD, that will be a service that will be something that people in academia can use for free, but will be a paid service for companies and businesses and things like that. So that is coming. But yes, we are hoping that the tool and the database itself are finalized in the coming months as well. Yeah.
okay, neat. And so one thing with the soil health testing, I guess that we talk about for this program now, one of the, I guess, features of the test is that they're supposed to be sensitive to management in the short term versus, say, looking at something like a soil organic matter test.
Mm-hmm.
So curious, I guess, for these indicators that you guys are looking at, how do you have a sense of how sensitive they will be to management? And it sounds like you're getting management data too,
Yes.
right? Okay.
Yeah, so the microbiome is very sensitive to management, and we've been able to see that even in 16S and ITS studies because we can see the community composition shifting with management. So we know it's sensitive, and it can be sensitive within weeks of a change. And so, yeah, the idea is that in the future you could be able to use these kinds of data to see if a producer was to make a shift in a management practice, that if you could take one measurement before and one measurement after even weeks, you could start to see if the microbiome is shifting in the right direction. So I think that's kind of like an exciting potential of where these kind of data can get to over time.
Okay. Neat. And we did have a question come through from Julian. Is this testing something that can potentially be financially reasonable for small farm operations, or would this type of testing be cost prohibitive outside of large operations, say thousands of acres versus less than 100 acres?
Sure. Yeah, so that is something that is very much at the forefront of our mind. We recognize that these kind of analyses are extremely expensive, and they are becoming more cost effective over time, but right now it's still somewhat cost prohibitive. So building this database is going to be really helpful because not to get too far in the weeds, but when you take these DNA samples and sequence them, one of the reasons why it can be so expensive is the analyses that it takes to go all the way through. But once you have a database, you can map your samples onto it without having to go through this long process, so it can significantly reduce the cost there. And I think as technologies are developing, we're realizing that there's a lot of ways that we can make it more cost effective. So I think right now it is probably cost prohibitive for a small farmer, but our hope is that in the next 5 or 10 years, we're going to see those costs really come down, and this is going to be something that is more widely used and can be available for a small farmer.
Well, thank you. And then we've had a couple more questions come through from Terry. Is there a geographic region or agronomic use, such as crop, pasture, forest, that your testing is limited to?
So we have primarily focused on cropping and grazing systems. We are not including forests in the data because, and I suppose if it was something like agroforestry, that would be cool. But as we've been collecting the data, coming from these different sources, it's been sort of tricky to figure out how to like filter through and find things that are relevant to what we want. So sticking to cropping and grazing systems has been the most practical way to move forward.
Okay, and then from Sean, did I understand that your testing is able to identify presence of nitrogen-fixing microorganisms in soil? And if so, are there indicators that their abundance has been historically underestimated?
Ooh, that's a good question. So yes, to the first part, it definitely will be able to test for those things, and I used nitrogen fixing bacteria organisms as an example because I think that they're an easy one to think through and they're pretty well characterized. I think there's a lot of other organisms doing other processes that are less easy to identify that will take time to figure out what those look like. But I don't know the answer to your second part if those have been underrepresented. But I would imagine for nitrogen fixing bacteria specifically, maybe not in the sense of I think they are more characterized. I think there's probably other functions that have been underrepresented because we just don't know which organisms are doing them.
All right, and then from Barry, in a pasture scenario, can you use organic matter level to determine fertilizer need, particularly in reference to north? Or, sorry, Barry, I'm not sure what the N means.
Nitrogen.
Oh, nitrogen.
Can you repeat that one more time?
Sure. In a pasture scenario, can you use organic matter levels to determine fertilizer need, particularly in reference to nitrogen?
Oh gosh, um, I don't know if I can answer that question. I think I'd have to think that through a little bit. I would say maybe. I don't know. I'm sorry. I feel like that might not be my expertise.
TBD, Barry.
I could get back to you though. I think I'd have to think it through a little bit.
And then from Jade, Oh, this is a wonderful question. In your research so far, or thus far, have you seen any correlations that surprised you?
Oh, um, that's a great question. Yeah, I think I think one of the big ones that has been that we've seen in the lab is that yeah, producers have there's been some, as I was saying, a lot of the nitrifying bacteria are pretty well characterized, and I think we've had a pretty good understanding of what they can do in the soil. And I think there's been some surprising results that have come out as to that microbes are often doing more than we realize. And so I think that's been one of the big findings is that, sometimes we take these approaches by thinking that like we have this understanding of what a microbe is able to consume or not consume or utilize, and then at the end of the day it's actually doing like so much more than we think. So I think right now one of the maybe surprising things has just been how much more microbes are capable of doing than I think our previous understanding has been.
Cool. All right, from Julian, is it possible to incorporate this information into fertilizer compost applications with the greater understanding of the microbial community's ability to immobilize nutrients?
Yes. Yeah. So I think for sure, and I think that's definitely the goal is to figure out how we inform management decisions using these kinds of data. And so that's why, yeah, getting back again to that, the nitrification example is that, if you were to see that your soil has this, that it's primed to be able to convert ammonium to nitrate where you could have leaching, then that can help you make some decisions on how you want to apply a fertilizer if you want to maybe use a nitrification inhibitor or split application. So yeah, I think there's a lot of different ways that this can end up translating into management.
And did you say if folks would like to keep up to date on what's happening at the lab, they can scan that QR code
Yes,
and
yeah,
sign for
that's
updates?
that's my LinkedIn. And so yeah, you're feel free to connect with me. I'm always happy to chat.
Okay, cool. Well, I don't see any other questions coming through the chat. So for now, anyway, you're off the hook, Laura.
Thank
Thanks
you.
for thanks for that. Yeah. So up next, we've got Jade from Ancient Grains. And it looks like you're ready to take it away.
All right. Let's see. Is that coming through okay?
Looks
Great.
great.
All right. So my name is Jade Koyle. I'm with Teton Ancient Grains in Teton, Idaho. And we, as you can see, operate from the Idaho side of the Tetons. We're not in the Teton Valley, but more over in the Wilford, Teton, Sugar City area. Um, and I grew up on a farm here in southern Idaho, and our farm was like many farms in the 80s and 90s. And we understood the importance on our farm of crop rotation, and we utilized that. Um, and I got a lot of mentoring and experience there from the family farm with my grandpa and my dad, and but eventually moved off the farm and didn't expect to get into farming. Until I came across some ancient grains in the early 2000s and started to explore those as a hobby. And it turned into a way to learn more about ways that we can provide food for our family. Looking at, just having grown up on the farm, saw the increasing dependence on chemicals and herbicides especially. And I thought, there's got to be a way that you can grow these things without that. And so it began as a hobby, more to grow grains for our family and for a few small customers, and it has slowly grown into something larger. And in that process, I began studying the organic program, which the organic program offers a lot of benefits and protections around what's not allowed in use of the crops, but I felt like there was a lot lacking in terms of, well, how do we solve these issues with fertility? How do we address the not only the economic side of the organic farming, but the fertility side of it? Because over time, organic farms tend to deplete and lose their luster over time on these organic fields. And so, as I started learning about this and exploring it, I was able to get some farm ground and start playing around with it. Again, this was a hobby in 2018, and my brother joined me, and my brother Jackson and I started, working on this together, and it grew into the operation that it is today. We don't farm a lot. We farm about 1,600 acres. Most of those acres are organic or transitioning to organic, and we use regenerative farming in that practice. And so a lot of the things that have been said today are directly applicable to what we're doing as an organization, and the benefits that we're seeing are playing out for us too. So some of the things that Dr. Vliet was talking about are this exact same questions that we've been looking to answer and explore. And the things that Laura's talking about are also very applicable. And what we're finding, and I guess going back first of all to the 2015 to 2018 time frame, I was studying regenerative farming pretty extensively, and I started talking with our customers about it, and I found that customers were very responsive to this. So if you think about, I think there was a question even asked about it, does it matter to the customers? And to the right customers, it does. And I started to notice that back then. So I started to talk about what we were testing on our farm, and what sort of results that we were seeing. And there was a lot of interest in it. But on top of that, we were seeing these results that were very interesting. Our basic program on organic was alfalfa as a rotation for cleaning up weeds, building up nitrogen in the soil, and then rotating into a year or two of grains. And we've now expanded beyond that to where we're always planting these grains, but we're planting different varieties of grains, including ancient grains, and we're planting different seeds in with them. So we'll have a tuber in there with them, or a legume, and it's this sort of a cycle, I guess, that if my slides will update, that we're trying to plow out in our operation. And to speak to each of these specifically, the soil fertility, obviously, building the fertility in the soil is multifaceted. In some cases, we are relying on some outside sources like soft rock phosphate that we bring in, and it slowly breaks down over time. And the higher our biology is, the more available that soft rock phosphate can be. Um, and and reducing the input of that soft rock phosphate over time, so that there's a larger availability of that made available through just normal soil breakdown and availability through the biology. Um, but the diversity of plant health, diversity of the plants that we're putting in is really important. So, for example, when we're planting these grains, we'll have a um, daikon radish, or uh, you know, one of the plants from that family, as well as legume. We've tested with quite a few different legumes, clovers, peas, and we're testing this to explore not only how it behaves in terms of the performance of the plants and the soil performance over the years, but also how well we can utilize that on the crop, on the food side of things. So in some cases, we're producing feed for animals. In other cases, we're just producing foods for our customers. With the end goal being here that if we put more attention into regenerative organic farming, we will end up seeing a more nutrient-dense food. And we are seeing results in our soils already. And I'll talk about some of those. We look at our approach to farming as you could call like a seasonal or annual, and then you have in-season practices. We look at our soil tests not so much as a read of what we're going to do today or tomorrow, but how we're doing overall year over year. And so for that reason, we're adapting our soil testing to try to get consistent information of how that field is improving over time. And one of the main markers we're looking at there is organic matter, um, water extractable carbon. And for the time of year, we're also looking at active biology and the rate or the abundance of that biology. So are we getting enough of it? But the things that we're using on an annual basis, regardless of how things are going, as sort of an active management, is cover crops, crop rotation, companion crops, compost tea, soft rock phosphate. These are the key elements that we use. And then as far as active management throughout the season, we're doing sap analysis, microbial tea, fertigation, foliar, and these are based on the sap analysis readings that we're getting, trying to adapt and and react according to that. We're struggling with sap analysis right now to get consistent results and reads on that. So we're also taking a lot of notes just based on how the plants look. Are they healthy? At the stage that they're at, are we starting to see the lower leaves or the coming production look different than the initial production? We're watching all of those factors on top of the SAP analysis to make sure we're holistically looking at how things are performing, because there's no doubt that the early season is different than the mid-season. And so we are using the foliar, fertigation, microbial tea to help support that. So we're using products like fish and other, certified organic products that are natural elements that the biology can break down. The microbial tea is important because we want to supplement as quickly as possible the active biology and help to activate it early. So we're covering our seeds or treating them with biology and then also applying it, as a fertigation or foliar. So these are some examples here of a field that we put in this year. In the center, you have the wheat growing with um, the clover, the crimson clover. Sorry, my mind blanked there for a minute. So you can see the red crimson clover out there, and there's lamb's quarter in there too. This is an organic field, so you've got weeds. And what we're trying to do is build a system that we have active cover and diversity so that the weeds don't have the need to express themselves as much. And this was a good example of that in this field where normally this was a third year grain field, but normally by that time you would see weeds become a big issue. But in this field, the weeds never flourished and they kind of stayed down low there. And that allowed the grain and the clover to express themselves, and they performed really well. The yield on this was better than the prior year, and so it's a good example of how we're seeing progress over time. Um, and then some of the active management that we're doing is trying to get the grains off quickly. We've been very fortunate to have these long fall with warm weather. The weather helps a lot with the cover crops. Normally we would see a frost in early September, but we didn't see that this last year. And so we grew really good cover crops this year, and we were able to get cattle on some of them. Cover crops with clover, peas, radish, alfalfa, volunteer grains, and that very diverse mix not only was obviously very good for the soil and the diversity that are brought there in terms of microbiology, but also the cattle side of it. Obviously, them coming in grazing. And we don't own our own cattle. We don't run our own cattle, so we worked with a neighbor on this, where we're trying to do more of it. And obviously, depending on how early that winter or fall frost comes, will determine how much of that we can do in the future. But trying to get the grains off quickly, get cover crops in, and start harvesting that sun, that fall sun, to try to take advantage of it and put it in the ground and use it as water extractable carbon for the next year. Overall, our end result and goal is nutrient rich and delicious food. That's what our customers come to us for. And we use all these different systems as part of our regenerative operation to try to help accomplish that. And for us, we see it as an aim as opposed to a method. And and what that means to us is that we're never perfect at it. It's always something that we're going to have to adapt to the current weather conditions. We're going to have to adapt to what's going on in the climate and the markets. How well we can market that. We're focused more on revenue and less on yield, more on nutrition and less on the or nutrient dense and the flavors instead of focusing on the amount of product we get off of an acre. And so what that ends up doing to us at the end of the day is saying, well, how can we raise more of our own nutrients? How can we mine more of nutrients out of the air, make more available out of the soil, and cycle nutrients better to make them more bioavailable to the plants and express themselves in the form of better foods. So we're really excited about everything, the research that you guys have been talking about so far on this presentation. It's directly applicable to what we're doing, and we will stand to benefit a lot by participating with you in that. And we want to support it and continue to pursue it. Our research is often very focused on practically how we can solve certain problems like weed issues or deal with compaction. That's why we use a lot of those tubers, or how do we help to bring more nitrogen into the mix? And surprisingly, one of the things that we're seeing is less and less reliance on the nitrogen. In the past, it was kind of the main question, and it's become less and less of the main question as our soils have become more balanced. There's much more nitrogen available than we expected. And so now the challenge for us becomes how do we optimize this system and get better at performing on it year in and year out? In the fields where we've had this running the longest, we're seeing a higher percentage of organic matter. Some fields that were in the low mid to high one that are now in the high twos, low threes. We've got some fields that even just a few years ago had some issues with compaction, and we're starting to see those issues go away by adding this diversity in and bringing the tubers in. So overall, if we can implement this system, the degree to which we get better at implementing it and reacting to the conditions and adapting and still moving forward with the plan and taking advantage of the natural resources there, that will help us determine how we get better returns on, the end product, the quality of the product, the yield, and eventually we see ourselves incorporating animals more and more into this, whether it's cattle or others. There's plenty of livestock people around here in ways that we can involve them in our operation. But it's not just hey, this is how we do it and this is the result. It's more of an aim and a methodology of whereby we're going to continue to improve and get better at this over time. That's all I had to present.
Awesome. Thank you, Jade. I'm gonna step in for Lucille for this round of questions as she had to step away for a minute. Thanks for all of that information and on your operation. I was curious, we have some questions coming in the chat, but I was curious if you could talk to us about your decision to grow and market ancient grains and kind of what that process looked like with acquiring seed and why you chose to grow ancient grains, if there's a difference between the nutritional density and content of ancient grains versus newer grains and that process.
Yeah, so ancient grains have interest in several different parts of the market. For me personally, I was just really intrigued by it, and it led to this hobby where I started contacting people all over the world, and people were reaching out to me. I put up a blog actually initially, and people were responding to that blog, and it sort of developed into these relationships with people who had seeds in different countries, and we were able to get those seeds. We maintain all of our seed programs today, but we were able to get those seeds by like-minded individuals out there who were caring for them, and we were able to get those and bring them into our operation. So it was, and every seed that we grow has a different story like that to it. So it's kind of an interesting journey and legacy that we've been able to be part of. Um, Now, as far as nutrient density in these grains, we have a Um, these grains are much lower yielding than many of the modern grains, and several of them even require hulling and processes that are not required with modern wheat. And so those could be considered drawbacks, but they also have advantages in terms of their ability to deal with disease and pest. There are plant scientists who are doing research on, for example, rust and how to prevent that or to mitigate it. And like einkorn is highly resistant to that, as an example. In terms of nutrient density, I don't know if it's because they're lower yielding or just inherently or both, but they tend to be higher in nutrients, higher in antioxidants. Um, they'll be higher in some cases higher in fiber. And so these are factors that our customers are looking for. They often know this already when they come to us. And so um, they know kind of which grain they want and why. Although we do some education about it as well. Some of them have a different type of gluten than modern wheat, and so that factors into it. But the markets are very small, and ancient grains have a history of boom and bust, and that is because of the small market. And so you'll see people come in, they'll grow a bunch, they can't get rid of it, and then they dump it on the market. And so we do deal with those cycles in our business, and we have to, stay focused on the long term and understand that some of that's going to happen. But overall, we're dedicated to serving the ancient grains market. And we see them as an important part of our regenerative operation, but also an important part of how we eat as a family and how our customers want to eat.
Wonderful. Thanks for that, Jade. We have some questions that have come into the chat. Barry's wondering, and this could be maybe a question for the larger group as well, is anyone using Redmond minerals either fed to their livestock or SR50 as a soil amendment to enhance soil health?
I'm not familiar with those.
And if anybody has a response to that, you can feel free to chime in or respond to Barry in the comments as well. Okay, next question. Do you monitor earthworm numbers from Nancy?
So we do dig in the soil. We look at the, are they there or are they not? We don't necessarily count them at this point, but their presence is important. And one of the big reasons that we don't use toxic chemicals is because it's fighting against us. And we wish we were no-till. We wish we didn't have to till at all, but we do have to till in some cases, and that ends up, fighting against us to some degree. But we feel like we're offsetting that by not putting toxins in as well, um, and we're trying to manage even the tillage part of it as best as we can.
Great. Carla's asking, how do you manage the high calcium carbonate content / pH in those loess soils in an organic system?
So one of the things that we found is that as we raise the organic matter percentage in the soil, the pH comes down. And so we've seen soils that were in the low eights come down into the high sevens, 7.6, 7.7. That is not easy to do, but and there may be other factors to that. There are people on here that are much smarter than me about these things that could probably answer that better. But that is one thing that we're trying to really focus our efforts on, is increasing the organic matter percentage, bring that pH down, and that makes the phosphate in the soil more bioavailable and other nutrients as well.
Wonderful. Okay, one from Lance. Do you swath your grains to get them to dry down before harvest?
So this last year we tested quite a bit with that, and we had a lot of success with it. Of course, the downside would be if you get a rainstorm, then you're probably going to be worse off. So there's a trade-off there. But one of the things with this regenerative system that happens is, you might have, for example, a radish in there and clover, and some of those might not be fully mature, or a pea, right? So the swathing is definitely an advantage if the weather permits it. We can, possibly cut off even a week there where we can get these cover crops planted earlier and allows it to dry down evenly, makes the combine run through it really quick and harvest a lot faster. We definitely saw that this last year.
Great. Thank you, Jade. All right. Kathy, we have a long question from Kathy in the chat. I can read it out. I have a 10-acre farm in the Wood River Valley where we had glacier leave us soil that is high drought-prone stone held together with clay and silt. My soils have every mineral except nitrogen. Does this type of soil have the same diversity and number of microbes as the soil that I am testing?
Mm. Yeah,
Um,
that's a very technical question. And there's probably better people to answer it, but I'll give it a shot. So we had, the bottom line is again coming down to bringing organic matter in there. And the best way to increase organic matter is to get something in there, a diverse set of species growing in there, and doing intensive grazing on it. If you could pull something like that on 10 acres over time, you would see some dramatic results. And so you want to have some intensive grazing and then the animals off of it so it can fully recover and restore. Um, and you can read different books about that. We tend to look at things as, what's the way that we can farm so that it will eventually work its way out. And so it's not like an immediate things you could do. There are probably immediate things you could do, like possibly putting calcium on there. And I'm sure there are other things that others can suggest, but ours would be more of a long-term management way of adding organic matter in there, helping get more oxygen down in there. The tubers would really help. But that plant diversity and intensive grazing, I think, would eventually heal that over time.
And she kind of mentioned that there was some more to the question that I didn't read. She's saying she's using composted horse manure and straw that's been decomposing and kind of asking about advice for a product that might raise microbial levels in your soil, maybe in her soil. Maybe something like I was going to ask you about the microbial tea that you
Yeah.
mentioned and whether you're brewing that yourself or kind of what that looks like. And maybe that could be something that could help Kathy on her journey.
Yeah, we have done it ourselves, and Shiloh Bingham's on here. He helps us with that as well. Um, but the microbial tea is something that is very inexpensive that you can build your own at your home. You'll get one of these 2,000 gallon or like pound or whatever they are totes. Those totes that are in a steel crate, and you can turn one of those into essentially a microbial tea bioreactor. A lot of videos online about it, and brew your own tea for very affordably. And that's a great way to put biology on. And of course, there are a lot of really good products out there as well that you can use to bring biology in. And I think that doing all those things would probably be smart if you want to get after it quickly, because using time, every season you only have one season. So as much as you can do to help that happen will only benefit you sooner, right? And you can multiply that, so you can say you got the biological tea, and maybe you plant your seeds with a biology on the seed, a coat on the seeds, and then putting diversity in there, and the combination of those all will definitely increase the biological activity in your soil. And I would
Wonderful.
I would just say too, when it comes to choosing cover crops, or what you're planting, study your soil type and study which plants, and because there are a lot of tools online about this, which plants are going to do best in the pH, the soil pH that you're dealing with, as well as the soil type. And that will help determine what plants you choose to help heal that specific situation.
Awesome. Thank you, Jade. Hopefully that helps Kathy out and other folks on this call as well. Great information. Um, that's all the questions I'm seeing for now. We can keep monitoring the chat. Oh, and Kathy says thank you. So that's great. Wonderful. Well, I think unless you have anything else to add, Jade, then we're ready to move on to Rem and Karman. Thank you so much for your time, Jade.
Thank you.
And Jade, if you want to stop sharing your screen. Oh, there you go. Perfect. And Rem and Karman, whenever you're ready, feel free to share your screen and go ahead. Great. That looks perfect.
There you go.
Hi, everybody. I'm Carmen Fox. This is my husband, Rem. I run Mad Fox Farms and do permaculture landscape design, usually for homeowners or for people that have smaller parcels of land, individual folks that really don't know a lot about all the topics that we're talking about today. So we do a lot of presentations that are very introductory for people who've never really talked, they don't really know anything about what is happening in their soil. And so we do designs for them because most of them are interested in bringing food into their landscapes, even if they have, maybe just a condo and they would like to just add a few things, but they don't know how to improve their soil without having to use chemical inputs. So we do a lot of training for that. And then Rem's side is he does nutrition counseling for people. So he is Iron Roots Wellness. And so this is kind of a overview of what our presentations look like when we're speaking to individuals who haven't really talked about a lot, haven't heard these concepts before. So here we normally start with basically what is the soil food web, always talking about the sun, starting that off, and how nutrient availability actually happens in the soil for plants. So as you can see here, we've got a base diagram of trees and how when they are going through their photosynthesis process, bringing down exudates, explaining what those are, basically desserts, carbohydrates, sugars, and whatnot that go down into the soil from the plants, letting the different bacteria and fungi know what they need. And talk to them about each one of these levels of organisms that most folks haven't heard of before, other than fungi. And bacteria, and how they are eating the organic matter, eating their soil, and make them think about how important that is. And then how each one of these other predator layers in your soil, if they're present, they're the ones that eat the bacteria, eat the fungi, ones that will be eating those different types of amoebas and ciliates, flagellates, all those kind of organisms, showing them how each one of these different layers are beneficial. Nematodes is always a good example because most people have heard about root feeding nematodes, not knowing that there are other nematode species that are beneficial by eating different bacteria and fungi, and how once those predators have done that, they are the ones that once they release their excrement into the soil is how the nutrients become available. Again, going back to talking about how the bacteria and the fungi are the ones that are taking all those nutrients in to begin with. And then as that process works with the higher trophic levels in the soil of different predators, how that actually comes to make those nutrients available, not only for the plant, but then as it goes on for all of us. And then we usually try to relate it back to, depending on where each one of our customers live or where we're giving a presentation, find which is kind of their keystone species for their area. And again, going through this same level, but then branching it out even further to help make it more manageable. It's difficult when you're talking to people about what's happening in the soil with microscopic organisms to really, have them mentally go, okay, well, how does that really help me? So we try to break it out into what is eating those organisms once they're not so microscopic. And so we talk a lot about how that leads on and why all of their poop is so important. So this is a good example of how that goes. And each one of these, as we're talking, will a lot of times talk to them about, how many of you have gone out and foraged your own, like in this example, huckleberries, or you've fished, or for people that, like in the leaf matter, hunt for mushrooms. And so trying to relate it to how they actually can use it in their own lives and how this is relevant to them before we really teach them how to start improving their soil and how important it is for their personal health. And then Rem will kind of take it over from here.
So these presentations so far have been exceptional. And one of the things, as a clinical nutritionist, if you look at why plant life is so important, why soil health is so important to plant life, there are a symbiotic relationship between plants and humans. If you look at just basic elements, nothing would be here without oxygen, hydrogen, correct? So if you think about that, plants themselves in most all facets have an epidermal layer, a reproductive system, a vascular system. They take in water, they present oxygen. They have nutrients. They have their own immune system, although it's an innate immune system. We as human vertebrates have a very complex immunology system. And the innate immune system that was mentioned earlier by one of the professors on the call talked a little bit about how some plants from an innate immune system can fend off predators, and healthy plants can do that as long as you can maximize their yield and their nutrient base and things of that nature, which is why soil health is so important. So we teach a lot of this stuff. From a toxin sensitive perspective, we are also, sensitive to toxins, leads and other things that can be presented in the soil. And of course, we're alcohol sensitive, and most people know that plants will die from alcohol poisoning. And so you have to look at some similar characteristics like that and really kind of associate how soil health and plant health harmonize to basically have us be more nutritious from the output of those plants. It wouldn't. There we go. So this is one example that we like to use in our presentation that talks a little about microbiology in the soil. And the soil itself, if you think about it, how do you get zinc into your body? And zinc itself is a non-digestible compound. You need actually a subsequent derivative of zinc in the soil, and how that is actually done is an example. Was brought up earlier with talking about earthworms and measuring worms. Karman is a soil technician. She actually, in part of her training, went through microscopic reviews of soil samples to take a look at all the different life forms that are in the soils on a kind of a per square inch basis, which is a tedious process, if you will, from a sampling perspective. But one of the things that we know about worms as an example, and if you use worm castings in any small farm or even backyard garden of substance, is that they actually produce an enzyme that actually break down zinc into what's called bioavailability. It was mentioned a little bit on the call earlier from some folks. Bioavailability is really important to plant life because we need bioavailability, which is basically the ease of digestion and how you can basically process it and put it into your body's cell structure for use to create energy. And so the more bioavailable you can make your soil to plant life, then obviously the plant life's health and its strength and how it can fully mature actually helps you with bioavailability and digestion. And you can get a lot of the nutrients that were mentioned on this call as well in that way. And we talk about those a lot. To give you an example of why regenerative farming was really kicked off in a lot of ways because of the natural leads and things that are in soils, and even from fertilization. And lead is not just added to fertilization. It can be a byproduct because of some of the industrial recycling. Once you have something like mycorrhizal fungi in the soil, it actually will act as a blocker, because if it takes up zinc into its structure, it doesn't have the homogeneity to basically not be more complex, if you will, taking in other metals. And so if you don't have these types of elements in your soil, as an example, where you have these life forms, you have higher risk of taking in toxic metals into the plants because it's easier. They have the pathways for it, so they will absorb it. But if you can really create these soil structures better, then basically is a great example we like to use with zinc is, it'll prefer to take it up, and that's how it gets in the plant. So you can basically eat it and have bioavailability for yourself.
So this is basically our contact information. Like I said, we do permaculture landscape design for folks, helping them utilize the resources they've got on their property. We're We're in central Idaho, and it's a very cold climate. It's a very short growing season. And so we teach people how to help improve their soil without, um, trying to avoid some of the difficulties that we ourselves have encountered with trying to work with such a short growing season
That's
to promote them growing food and native plants.
It's tremendously hard on even 5 acres, let alone the 1,600 that Jade is facing. So really appreciate your effort on what you're trying to do. That's
Yeah.
for sure.
Well, thank you so much, Rem and Karman. So cool to see that you guys get to work together with your different expertise. That seems really special. And good to also see where you start. The soil food web diagram that you shared, that's often where Courtney and I start when we're doing a rainfall simulator demo. So for any audience, I think a good reminder, like you said, that it starts with the sun. Curious in your work with folks, what ideas or themes seem to resonate most with them as you're introducing them to all of this?
One of the things that a lot of folks will come to us because maybe they've just built a home, or they've got an area on their property that is problematic, like with weeds. And one of the things that we try to encourage, kind of like Jade was mentioning, with trying to determine what is the best kind of cover crops for people to put in based on what kind of weeds they already have present that they're trying to get rid of. So we try really hard to teach people to kind of do some of their own investigation. If you're growing a weed that is, in the aster family, and it's doing really, really well, what other plants that are in that family could be native to your area that you could try seeding? Because chances are what those weeds are telling you is that I really like the conditions of the soil. The bacteria and fungi levels are good for me at this, I'm obviously growing other plants in this family. So how to introduce plants in the same family that you already know can thrive from those soil conditions, but ones that are beneficial in that family. And so that usually is one of the biggest things that people just look at you like you just hit them in the side of the head, because it seems very logical. But we actually had listened to a permaculture podcast where people were doing a lot of research on that. And so we've tried it on a couple of places where we have some sketchy plants that are trying to make a go, and it has worked out really well. And so that is one of the things we try to implement. I think also the nutrition side of it. I'm surprised how many people don't link their actual, if what you're putting on the plant, how that's going to in turn, work for your own system, and the relationship between the two sometimes.
I think
Feel free to-
the big market of growing organic names and organ health and things like that, people feel like they want to have a healthier outcome. That's always been a big issue. In small farms that are being developed, if you build a new house on 5 acres and you have growing aspirations, starting is what people just really struggle with. Depending on where you live, especially in Idaho, we're north of Boise by a couple of hours. Super short growing season, terrible valley floor, was in a lot of over fertilization characteristics in the 40s from potatoes, had a lot of cattle ranching, has a lot of minimal water challenges with regards to that, but the soil is heavily in alkali. And how do you change that composition for people to get started to grow their own food? Because a lot of them want to have an interest, even on a small scale, of homesteading. And they always look to, do I do a greenhouse? Can I do anything outside? How do I do an edible landscape with, native plants that are, in the berry family, et cetera, and just kind of create more of a natural food forest environment? That's why a lot of people come to us, and we help them kind of create that. But it starts with that whole soil ecology study to really understand where they're at and how much aspiration they want to take on for the work level and get the results is kind of where we always start.
Yeah, I imagine with all those considerations, it can be overwhelming for folks who are getting started. I guess in addition to working with you, are there resources that you like to point towards people for? to increase their
We
learning?
yeah, we actually do recommend the U of I extension services because you can do soil testing, right? Exactly. Um, and there's a lot of great resources as far as helping people understand if they want to know what is in their soil to start with, especially their level of organic matter and where their pH is, kind of going back to what Jade was saying. And helping people see if if you have a specific amount of a certain plant you want to grow, like blueberries, like at our home we had a a very low amount of organic matter when we very first started, and we've slowly increased that, and we've had the same results, bringing down that it brings down the pH to where we now successfully grow a blueberry crop for us. And being able to have the proof that those things kind of work. We do recommend the first soil test because people like to know where they've started at least and know what's in their soil. And then showing them different ways to be able to increase their organic matter, increase the biodiversity of the organisms in the soil. We recommend a gal that's actually in Emmett, Big O Worm Farm. She does, Anita. You can buy worm bins from her. You can buy just her worm castings. And then we instruct people on how to just make a simple extract with that to be using for themselves. And I'm trying to,
It works as
I know, I'm trying to think who else.
well, the compost tea for that on vermiculture is really helpful. It also works as an inoculant and a lot of plant life as
Yeah.
well. So it's not just soil.
Yeah, I actually met her at an event a couple weeks ago.
Yeah,
And it
she's a
sounds like she
great
also
gal.
does,
Yeah,
yeah, she
she's
does
a great
educational
gal.
events too.
Yeah, she's a
Um,
great gal. We have some seed companies that we use. Seed Trust is one that they used to be in Sun Valley. I'm not even sure if the original owner still owns it, but they do a lot of cold crop seeds that they have tested for vegetable gardens that can stand, a little bit cooler temperatures. We've ordered from them quite a few times. And then we're going to be ordering from a company, I'm not gonna great. I think it's Great Basin Seed out of Utah, because they have a lot of really good mixes that for cover crops that we're wanting to look into for fire mitigation. We work with the Valley County Fire District, which not fire department, but the ones that help with grants. If you are a community up here, like a subdivision that needs mitigation done for fire management, and so they've asked us to try and find a source for seed for people to replant so that they don't have weeds come into an area after they've had mitigation and the soil disturbed. So fire
okay.
working group.
Yeah.
Yeah. So
And we did have a question. Looks like Courtney answered it.
Yep. Big old worm
Yeah,
farms in Emmett.
yes.
Yep.
perfect. Okay. And then you said something, and I think Jade said something about this as well, that the process of lowering the pH takes time, right? It's a slow
It
process.
does.
How long did it take you to get to the point where you could grow blueberries?
Well, when we finally started like, oh, this isn't going very well, and focused more on it, I would say it took us two seasons to really get the soil where we needed it to be. So then on our third season would be when we had our first good crop. And we used not only cover crops, but we also did like the hugelkultur bed method, but more just the layering. We're not super big on the actual percentages of, sticks and leaves and, the compost, and, we kind of throw whatever is in there that we've got that's already slightly breaking down and layer it, and doing that for two seasons with just a basic mix of a cover crop that did have some clover. It did have some radish, and it had was it a wheat? It had a wheat in there too. I'm not sure the variety. And so then mowing that down Um, we did also one time use chia seeds because they frost off
Oh, neat.
really quickly, and that also seemed to help getting that under control. So we try to use
Okay.
the least intensive processes where we're just letting the organic material do its composting for us instead of I'm horrible at making compost. I'll admit that it's really tough where it gets hot really quickly and trying to keep it the right moisture and whatnot. So we've had more success with doing the layering, and we've had more success with adding the worm castings and cover cropping.
Mostly that's due to, if you look at Courtney's background in the snow, when you do the hugel beds, you get the snowpack. And so when you have 4 feet of melt off over the winter on all of that, your composting accelerates because of the pressure of the weight, the water, and the melt off and everything. And
The
so
warmth that's
that really helps in the higher elevations for sure.
Okay. Neat. This is great information, I imagine, for our audience today. We also had a question come through from Barry. He asked, what pH values were you dealing with?
When we started, I think it was a little over 8. We had a couple of spots where, typical, you dig down. Our house was older, had been on the property for quite some time, but other than the lawn that they had put in, a little strip of lawn, there wasn't really, it was all mainly just natural grass that only grew a few inches each year. It's roughly 2 to 3 feet. And we've mowed that down a few times for fire suppression when we've had some fires getting pretty close and left that. And so we now, I'm not sure, we didn't test this last year where we were, but we had finally gotten to 7. So I felt like we were pretty good pH when we got to 7, but that would have been a year ago. So I'm not sure what it is. This last year was when we finally started getting the blueberries. So I think by the time that was in the spring when we had done it last, well, I say last year, but I don't mean last year. I mean, not last summer, but the summer before. So 2024 is when we last tested and had gotten it down to 7. So in the berry patch area. So I'm not sure what it is from last growing season.
Well, we had a comment from Jade. Thank you for your presentation. 8 to 7 pH is impressive.
It was. I was impressed.
All right. Well, with that, I am seeing no other questions coming through the chat. So I think now is a great time to wrap it up unless anybody else has any last minute questions that they would like to get in for folks. And it looks like we are not getting any. So I'll go ahead and we'll end it here. So thank you so much to everyone who joined us today for this last 5 for 5 of this season. So we'll start these up again probably in December of this year. So please, if there are folks that you would love to see featured on the Idaho Soil Health 5 for 5, or if you would like to be featured on the Idaho Soil Health 5 for 5, please feel free to reach out to me and Courtney. And again, today's webinar will be available on the University of Idaho College of Ag YouTube here in probably the next month or so. So thank you so much and wish you guys the best of the rest of this winter season.
Thank you so much.
All right. Bye.
Thanks, everyone.